Welcome, everyone, and thank you for joining us today. I am Perry Eker, and a scientist at HNRCA. I want to remind this is our last seminar for this season, and we are lucky to have our speaker, Dr. Rio Sanabria. Um, Dr. Sanabria is an assistant professor at the USC Leonard Davis School of Gerontology. They earned their PhD from Columbia University and completed postdoctoral training at UC Berkeley before establishing their lab at USC. Dr. Sanabria's work investigates how the actin cytoskeleton coordinates cellular stress responses across aging. Their research spanning C. elegans, mammalian systems, and emerging non-model organisms have been recognized with major awards, including an NIH Pathway to Independence Award, a Stephen I. Katz Arrow One, and support from Glenn and Hillblum Foundations. Dr. Sanabria is also a dedicated leader in geoscience training and institutional service. They co-organize the Geoscience Los Angeles meeting, serve as an associate editor at Geoscience. They also have chaired influential cultural affairs committees at USC that advance mentorship, equity, and community. Today, Dr. Sanabria will present more than a loading control, studying the actin cytoskeleton for novel anti-aging mechanisms. Before we start, a quick note for the attendees. Please submit your questions through the Q&A section. And Rayo, thank you again for joining us today, and we are excited to hear your talk.
Great. Thanks very much. I'm really happy to be here to share our research with you. So thanks to you all for being here and thanks for the invitation. So as Perry said, my lab is really interested in studying the actin cytoskeleton, which I'll introduce in a second. We study aging from a very fundamental cell biology perspective, and I know this is a nutrition seminar, so I'll try to sprinkle in as much nutrition as possible. We also study the mitochondria a lot, which is where a lot of our nutrition work stems from. We are a multi-model system, so you'll see these little icons on the bottom left corner for any slide that has data from our lab. And so you'll know which specific model system I'm talking about. I will try my best to remember to tell you every time I switch model systems, but I have a tendency to forget, which is why I have these little useful icons. So what is actin? Actin is a really important part of the cell. You may remember from your high school or your introduction to biology college lecture about all of the organelles and other components within the cell. And what you'll notice in a lot of textbook depictions of the cell is that the actin cytoskeleton is often missing. So are the microtubules, and usually the centriole is the only cytoskeletal component that's listed in these cell biological schematics, which is really a crime because if you actually look at what the cytoskeleton looks like inside of a cell, so this is a mammalian cell. This image is from Andy Moore at Genelia, and you can see that looking at the actin cytoskeleton, actin is essentially all over the cell. It's one of the most abundant proteins inside of the cell, usually tying for first place with ubiquitin, depending on the cell type. And not only is it one of the most abundant proteins, but you could see that it basically has these really fine ultrastructural components. It makes these really nice filaments. In some places, it makes much more aggregated looking things. It makes a lot of barbs at the end of the cell membrane, and then it makes a lot of really intricate structures throughout the entire cell. So considering that this is one of the most abundant proteins and the cell needs to utilize a lot of energy to first synthesize it and then organize it into these structures, you would imagine that it's a really, really, really critical part of the cell. And so actin is also unique in that its regulation is not just in terms of synthesizing a protein. So step one is synthesizing a protein, the monomer, which is called G-actin. And then it has to be synthesized into these filaments or polymers called F-actin. So I say it's very analogous to like a brick or a stone, where maybe a single brick or stone doesn't have a lot of function. But then you can take a lot of bricks, put them together into really nice intricate structures, such as creating a highway, structural scaffold for your house, or something like that. In the same way, G-actin doesn't have many functions, but it can be polymerized into a lot of intricate structures into these F-actin filaments. And that structural synthesis into these filaments is regulated by a handful of proteins. And I'm just going to show you the major family of proteins that regulate this. There's definitely no need to remember this. It's just kind of a highlighting that actin is not just a single protein that makes up up these polymers, but it's actually a multi-protein complex. So we have proteins like the arp 3 complex, which synthesize filaments into these kind of like branch-like structures like a tree. You have foramins that synthesize these single filament structures. You have cofilin that kind of pulls apart these subunits and allows you to recycle actin subunits. And then you have actin binding proteins like tropomyosin that can bind and stabilize it. So again, you don't need to remember what all of these cell components are, but suffice it to say that there are over 100 different proteins that regulate this process between this single monomer to this polymer form. And so there are a lot of proteins that have to work together in a concerted effort to regulate the dynamics of actin. And all of these different regulatory components work together to allow for actin to have really diverse functions, such as providing structural support for the cell, trafficking, that's like moving things from one end of the cell to the other. Anything that has to do with motility or cell division is regulated by the cytoskeleton. And then in the last few decades, it's been really understood that actin plays a role in a lot of different other processes, such as nutrient signaling, where it feeds into really major nutrient signaling pathways like mTOR and AMPK. It's really important for regulating organelle dynamics like mitochondrial fusion and fission, transcription, stress response, DNA repair. Really, any process in the cell that you could think about, the cytoskeleton is probably involved in one way, shape, or form. So not only is the actin protein itself really challenging to regulate from a molecular biology standpoint, but then this actin polymer itself needs to regulate all of these dynamic cellular processes. And then if we zoom out a little bit and think about physiological functions of actin in terms of what it does inside of the human body, you may remember again from a basic anatomy and physiology class that actin is one of the two filaments along with its counterpart myosin that regulates muscle contractions, where you have these really nice actin filaments and then myosin motors combine to it and pull, which allow for muscle contractions. They're really important for the gut barrier. So your intestine has a lot of bacteria inside of it that you don't want it to leak out. And so actin really holds intestinal cells together to prevent the bacteria from escaping the gut. It's really important for neuronal signaling. So there's a lot of cytoskeletal components in the brain that allows for secretion of neuropeptides, hormones, etc. And then it's really important for wound healing, where if you have a cut or a scrape, actin really regulates the cell migration and also the cell division that allows for wounds to close. And what's important is that all of these processes that we think about of what actin plays in the physiological body, all of these things deteriorate during the aging process. So we could think about as we age, we lose muscle function, or we start to get leaky gut syndrome. We think about dementia, where neuronal function is reduced, wound healing slows down. And what my lab is trying to do is understand how age-associated breakdown of actin can contribute to the physiological breakdown of many of these aging pathologies. And so in this way, we're trying to argue that actin is potentially a biological hallmark of aging. So for those of you who aren't familiar with the biological hallmarks of aging, all these aging scientists came together and tried to define what are the things that happen on a fundamental cell biological level that drive the pathology of aging. And these 12 components were essentially put together. And, every few years, there's more and more and things being added to this wheel. But we can think about some of these things that are very, very, very well established. For example, genomic instability. We know that as we age, things like exposure to sun and radiation can increase DNA damage and DNA mutations. And accumulation of those DNA instabilities can actually drive pathology of aging. We may all be familiar with the mitochondria, which is really important for energy homeostasis. And as mitochondrial function declines during aging, then we start to accumulate aging pathologies. And what my lab is trying to argue is that actin dysfunction deserves a position on this wheel, because similar to all these other hallmarks of aging, actin dysfunction could be a direct driver of aging pathology. So to argue that something is a biological hallmark of aging, we need to show three things. First, that actin deteriorates during natural aging. What that means is as an organism ages naturally, normally throughout its lifespan, you should see that actin deterioration occurs in old individuals. And then you have to also experimentally show that experimental targeting or disruption of actin can cause or induce aging pathology. Whereas if you protect the cell from actin dysfunction, that is, remove actin dysfunction and make actin properly functional in an older individual, it should mitigate some aging pathology. So my lab kind of sought to define that actin does follow these hallmarks. In terms of human aging, why do we think actin could be important? I told you that actin plays a role in all these physiological processes, but then if we look at specific age-associated diseases like Alzheimer's disease or neurodegenerative diseases, cytoskeletal abnormalities is already one of the hallmarks of neurodegenerative diseases. And for those of you who are familiar with Alzheimer's disease, Parkinson's disease, Huntington's disease, and all these neurodegenerative diseases, one thing that's known about them is as you age, the risk factor of getting these diseases increases exponentially. And the reason is because a lot of these hallmarks of neurodegenerative diseases are shared directly and overlapping with aging hallmarks. So as we age, a lot of these things start to happen, which increases your risk for Alzheimer's disease. And similarly with things like muscle myopathies and cancer, as we age and a lot of the things that occur during aging, a lot of the things that break down in the cell during aging can be drivers of these pathologies. And actin dysregulation and disruption are direct drivers of many of these age-related diseases, which kind of adds a little bit more evidence that perhaps studying actin during the aging process is an important thing. So kind of to like zoom out on a really population level to see if actin is important for general aging, not disease-related pathologies, but also just aging in general. I paired up with two demographers, Arpa Wong and Eileen Crimmins, and they do a lot of population health studies. So population health studies are really, really, really important because what it does is it allows us to look at a very large population of humans and see what specific genetic risk factors exist that can be associated with specific aging hallmarks and aging pathologies. So we do something called the genome-wide association studies. What that is we look at single nucleotide polymorphisms or SNPs. So what SNPs is that our genome is about 97 to 99% identical, depending on where in the population we're from. And in that small difference that we have between our own DNA makeup, those are called single nucleotide polymorphisms. So that means that in a specific gene, I might have an A for A nucleotide in a specific locus, and you might have a C nucleotide. And what we do is we look at these differences between each and every one of us and see if these differences could potentially put you at risk for a specific pathology. In this case, we're looking at aging pathologies. So we did this SNP analysis. So what you're looking at here on this plot is the y-axis is p-values. So each of these lines is a p-value cutoff, which the thresholds are shown there. And each of these dots represents a single nucleotide polymorphism. And so we're looking at specific aging pathologies here. I decided to focus on muscle function because I told you that actin is really critical for proper muscle function. And you can see that there are four SNPs, two that are associated with decline in gait speed and two that are associated with change in frailty index. What that means is if you have a particular SNP, you will be at much higher risk to develop these specific aging pathologies at a younger age. So just by having a single nucleotide difference, and what I should have told you is that we're looking at the actin genome here. So we're looking at the actin gene locus, so our specific actin gene. If you have one specific nucleotide, it could put you at a much higher risk for having a premature breakdown of muscle function, which is really important to think about that a single DNA difference, a single base pair difference, could be a major driver for a specific aging pathology. If there are any demography, public health, or epidemiology people in the audience, you may look at these p-value cutoffs and you'll see that I used a biology p-value cutoff of 0.05, which, our ends here is looking at 10 to 20,000 individuals. So that p-value is actually considered quite weak in these large-scale demography studies. So to see if this is really significant, we decided to do a replication study where we looked at a different cohort. So this cohort was an American cohort and this cohort is a European cohort. So very, very, very different individuals. It is primarily white. A lot of these large-scale demography studies do have a tendency to be white skewed, and that's just because of who winds up being volunteered for these studies. But we did basically the same exact genome-wide association study, and what you'll see here highlighted in yellow is that two of our SNPs that we identified in the American cohort were also identified in the European cohort. And so that was really exciting for us because we now can identify these specific SNPs, that is a single base pair difference between each of our genomes, where having one SNP could put you at a much higher risk factor for having a decline in gait speed, that's walking speed. So reduction in skeletal muscle health during aging. And this is just one single base pair difference in the actin gene. So this is not a major fundamental loss of actin function or something. This is not like a mutation that can disrupt the entire function of that actin protein. This is a single base pair change, which will change one single amino acid and maybe skew the actin protein to be a tiny bit dysfunctional compared to a fully functional actin. So just by losing that tiny little percentage of actin function, you could just be put at a significant risk factor for something like skeletal muscle loss during aging. So that was really exciting for us. And while these population studies are very exciting because this is in human data and we can identify human risk factors, this is all associative data. It's not causative data. So this is a correlation and not a causation. And so in biology, we really like to look at causation. And in order for us to look at causation, that is, if I induce actin dysfunction, will it cause aging? I need to move to model systems because, of course, I can't go in and disrupt actin function in people unless any of you are willing to be a volunteer for that. So we decided to turn to C. elegans as a model system. The reason why we like C. elegans is because most of the aging mechanisms are conserved, and a lot of anti-aging interventions were first studied in C. elegans, identified in C. elegans, and then translated into mammalian systems. So it is a really great model system for aging. It's also very short-lived. It lives for about 3 weeks, so you could do an entire aging study in 3 weeks as opposed to 3 years in mouse models. They're very inexpensive, so you could probably do anywhere from 100 to 150 experimental interventions in worms for the same price as what it would cost to do one single intervention in mice. So that's really, really scalable if you think about large scale genetic or chemical compound screening. The other thing that I like as a cell biologist is that they're small and they're clear. So you can actually mount a C. elegans onto a microscope slide, put it under a microscope, and image intracellular structures in a living, breathing organism. Worms are one of the only multicellular systems that you could do this. You can't do this in flies. You can't do this in mice. In larger model systems, you have to actually dissect the animal in order for you to be able to see their insides, which means that you're introducing some artifacts with fixation and stuff. So just to give you an example, here's looking at the actin cytoskeleton in the entire lifespan of a worm, starting from egg to all the larval stages into adulthood, young adulthood, middle adulthood, all the way basically until death. So I can visualize the actin cytoskeleton in a living animal from birth all the way until death. And C. elegans are really the only model system currently that you can kind of do this in. And so I'll just give you a zoom in the actin cytoskeleton and what we're looking at. So we're looking at here actin in the body wall muscle. So the body wall muscle of the worm is equivalent to what the skeletal muscle is in people. And you can see that actin make these really nice organized linear striations. And these are lined up directly along the myosin filaments that I told you about earlier, which allows for muscle contractions. And if you look at the muscle actin during aging, you could see as that animal ages, you could start to see that the disorganization of actin starts to occur, where they start to get a little bit squiggly and disorganized until eventually it completely breaks down and you can actually see breakage of actin filaments. This time point when you actually see disruption of actin is when muscle function is disrupted. So once the actin in the muscle starts to deteriorate, that's exactly the time point that you start to see muscle failure. And if we image the actin cytoskeleton in mammals such as mice and look at actin throughout the aging process, it looks pretty identical where you're going to see these nice linear striations that start to break down. And the breakdown is directly correlated with the time point in which muscle function is lost. So this really shows that not only can we very easily visualize actin throughout the aging process in worms, but that it is directly translatable to a mammalian system. I'm just showing you muscle here just for simplicity, but we did do this in the neurons, the intestine, the skin cells as well, and we see exactly the same thing happen. So that's still correlative evidence to show that as you age, actin breaks down. And in order to show causation, then what we would need to do is you have your normal wild type animal, and then we break actin. So if we break actin, we should start to see essentially pathologies of aging. So it's essentially inducing destabilization or breakdown of actin, and then do we see pathologies of aging? So we did something really simple here where I told you about all of these different proteins that regulate actin in a lot of different ways. So in worms, we could do RNAi knockdown, so RNA interference to get rid of the expression of these proteins very easily. So what we did is we just did targeted knockdown, get rid of some of these components to essentially break actin. And then we looked at lifespan. So as I said, we did RNAi knockdown of these specific components. Those are the things that regulate the building and infrastructure of actin. So we're just essentially breaking actin. Um, and if you're not used to looking at a Kaplan-Meier curve, what you're looking at is percent survival on the y-axis across days on the x-axis. So here in blue is our wild-type curve, where as you go longer and longer in days, more and more of the population dies down. We have a median lifespan of about 17 to 20 days in C. elegans, as I said earlier. And you can see that if we do targeted disruption of actin, we result in a significant decrease in lifespan. So that's that curve, lifespan curve shifting to the left, showing that the entire population is living shorter if we disrupt actin. And of course, when we think about healthy aging, it's not just lifespan. So it's not just living longer or living shorter. It's about being healthy during the life that you have. So we did look at metrics of health span as well, which in worms, we usually look at reproductive health, muscle function, intestinal health, neuron function, and wound healing. And I'm not going to show you all of this data, but essentially, if you disrupt or break actin, you also show a reduction in all the health span metrics that are measurable in the worm. So not only are they living shorter, but they're also less healthy if we disrupt actin. And then because I'm a cell biologist, I wanted to see like if we disrupt actin, do we induce cell biological features of aging? So can we see these biological hallmarks of aging be exacerbated? And if we're right in that disrupting actin is actually inducing aging and not just making the animal sick, then we should see actual biological hallmarks of aging start to appear as well. So, the example of this is like if I take a hammer and smash somebody in the head with a hammer, I'm not making you age faster. I'm just making you damaged and sickly. So, of course, you're going to live shorter. Whereas if I actually go into your body and let's say that I induce sarcopenia in your body, then I'm not just making you sick. I'm also making you appear more aged. So that's what we're testing here. Am I just breaking actin and making animals sick, or am I actually inducing aging? And so that's what we wanted to look at. So I'm just going to show you a few different examples. So here we're looking at mitochondrial morphology. So this is a mitochondria targeted GFP. So what you're seeing here in green is the mitochondria. For people, if you are a worm person, this is a hypodermis. If you're not a worm person, that's kind of like what the liver is for the worm. And you could see that mitochondria make these really nice, like kind of like finger-like structures. We call them mitochondrial tubules. So in a healthy, normal cell, you get these nice tubular mitochondria. And then if you disrupt actin, you can see that mitochondria fragment. So fragmentation of mitochondria is considered a bad thing. These mitochondria are not functional. They don't respire. They can't produce ATP. They're not utilizing oxygen. So just by breaking actin, I cause a very, very, very severe mitochondrial fragmentation phenotype. You'll probably see that in this one specific case, you don't see mitochondrial fragmentation. You actually see a hyperfusion of mitochondria. If for those of you who are interested, this is inhibition of tropomyosin, which is what stabilizes actin. So we were actually intrigued to see this. One may think that because fragmentation of mitochondria is bad, hyperfusion of mitochondria might be good. But if we actually look at respiration capacity of these animals, they also are not respiring. So this is a very dysfunctional state as well. It's just kind of in the opposite direction. And we're still trying to figure out mechanistically why that's happening for that one specific inhibition. Um, we also decided to look at a bunch of other phenotypes, and I'm just going to show you like lipid droplets because this is a nutrition crowd. So one thing that's really important in worms is that maintenance of lipid droplets is their way to maintain energy and essentially ensure that they can live a healthy lifespan. So having a large number of lipid droplets in the intestine is directly correlated with being healthier and having a longer lifespan, whereas reduced lipid droplets in the intestine and reduced ability to carry and store energy is considered a bad thing. So this might be a little bit counterintuitive when we think about human health, where having more fat might be considered bad. But if we actually think about the evolution of humans, being able to store fat was actually a good thing that allowed you to survive famine, that allowed you to survive starvation. So this evolutionary benefit of being able to carry fats and store fats better is kind of like what flipped us to be bad in a Western diet when food is plentiful. So in C. elegans, again, having this ability to store lipids is actually a good thing. And you can see that in the conditions where we disrupt actin in any way, you see a massive reduction in lipid droplets. Some cases you actually see a reduction in number of lipid droplets. Some cases we see a reduction in size of lipid droplets. And then in some cases we see both reduction in size and number. So actin is really important for maintaining these lipid stores, and you can actually cause premature dysregulation of lipid homeostasis and lipid storage and essentially induce lipid storage disorders when you disrupt actin. So I just showed you those two things, but we assayed essentially a bunch of different things like protein aggregation, autophagy, gut barrier integrity, dysbiosis, essentially all of the features that you would think about when you think about healthy aging. And you could see a lot of these down arrows here, where in most cases, we actually see a disruption of things associated with aging anytime we disrupt actin. So this is all work that Maxime did for his PhD thesis. And what I've shown you so far that at least in worms, actin shows age-associated deterioration during the aging process, and then directed disruption of actin can actually drive premature aging phenotypes, such as these biological hallmarks of aging starting to appear, reduce physiological health, reduce health span, and reduce lifespan. But everything I showed you so far is in worms, so then it kind of makes you think like, well, if this is a worm specific thing, like, is this really useful for like general aging? So the first thing we sought out to do is, okay, is this a worm specific thing, or do we see this in non-worm model systems as well? So one of our collaborators, Ted Schmidt in David Walker's lab, decided to do a very similar thing, but in flies. And he's a neurobiologist, so he decided to focus on the brain. So what you're looking at here is a Drosophila brain, and they do a really good job of dissecting out the brain and then just visualizing actin during aging. And so this is a very zoomed out image so that you can see the whole brain. This is using an actin dye called phalloidin. So phalloidin will bind to the filamentous, that functional actin I told you about earlier. And you can see that in the fly brain, you see an increase in this phalloidin staining, which suggests that there's an increase in filamentous actin with aging. Now, you might think that filamentous actin is the functional actin, so having more of it is a good thing. But if you actually zoom in and do high resolution microscopy, you could see that the actin makes these big blobs or these balls, and these are aggregates of actin. So actin should not be making blobs and balls. It should be making like linear filaments. Like if you really look at certain parts, you could see these lines of actin. That's what you should be seeing. And these big blobs of actin are not normal. And so you can see that as the animal ages, you see a massive increase in these actin rods or aggregates, which are also called rods. And this aggregation of actin can actually drive neuron degeneration and neuron death and neuron failure. And if you resolve these aggregates, you can actually mitigate aging in Drosophila. So this is a really interesting thing because it showed us that in other model systems that do actually have a functional brain, in C. elegans, they have neurons, but they don't have a brain. You actually see an age-associated increase in actin aggregates, which can be a driver of aging pathology. And this is a really interesting thing because these actin aggregates are also seen in neurodegenerative diseases in humans, like in Alzheimer's disease. And I'll show you that in just a second. So another model system that my lab is currently developing is tardigrades, and I'm not going to talk about this that much, but I could give a whole lecture on this because it's been our obsession for the past few years. Why do we care about tardigrades? They're a really interesting model system. They are also called water bears. Some people call them moss piglets because they essentially live in the water. They live in an aquatic environment. Anywhere that there is moss, lichen, algae, those are its food sources. There will for sure be tardigrades. So just like you will see C. elegans in any dirt environment or in the mud, anywhere there's bacteria or decomposing food, you will see tardigrades equivalently in any hydrophilus, that is an aquatic environment where there is greenery. So essentially anywhere there is water and sun, there will for sure be tardigrades in there. What makes tardigrades a really unique model system, though, is that they have the ability to enter into something called cryptobiosis, where they basically release all of the water from its body, completely shut down metabolism. And when they're in this cryptobiosis state, they are extremely stress resilient. So I kind of say it's analogous to like a grape. So if you have a grape and it's like, a normal fresh grape, if you drop it on the floor or if you step on it, you'll smash it and destroy it, right? But if you dry out a grape by essentially removing all its this water and turn it into a raisin, you can kind of throw it against the wall, pelt it on someone's head, maybe step on it a little bit, and it'll still be fine, right? Because raisins are a lot more resilient than a grape. So that's what these bears do, where they turn into this cryptobiotic state. They become super stress resilient. You could put them in the oven, you could shoot them out into space, you could put them in a -80 freezer, and they survive. So that's a really interesting phenomenon for somebody like me who studies stress and aging, of trying to understand what allows this animal to enter into this cryptobiotic state, what allows them to become hyper stress resilient, and can we utilize some of these methods to essentially understand how we can make our own bodies more stress resilient so that we can utilize, for example, things like cryopreservation of tissues for transplants or cryopreservation of our entire body if we wanted to do long-term space travel. And that's really where tardigrades as a model system shines, of understanding how this intricate cell biological system could be used and translated into people. I'm not going to talk about any of that. Instead, I'm going to focus on actin because sometimes I can stray away in tangents too far. So here's what actin looks like in the tardigrade. And you can see that they have really nice structures of actin. What these bars are that you're seeing are their muscles. So tardigrades have these really thick actin fibers, very similar to insects, actually. Um, and I'm not zooming in, but if we zoomed into one of these muscle tissue, you would be able to see those nice linear striations similar to what I showed you in worms before. I wanted to do a zoom out so you could see the entire bear because I think it looks really cool. And here's a young, healthy animal, and here's an old individual. And you can already see very clearly that there are pockets where the actin organization is disrupted, where you could see filament breakage. You could see parts of the body where it looks like actin is starting to aggregate. You could see parts of the body where, like, there should be filaments of actin, and now they're just turning into these balls or little kind of nuggets of actin. And so, much like C. elegans, throughout the whole body of the tardigrade, you can see that there is for sure a deterioration of actin with age. And so, does actin show deterioration during natural aging? Yes, I showed you in worms, flies, and maybe tardigrades. Yes, I haven't shown you in yeast, but in yeast, it's been shown that actin also deteriorates during the aging process. But these are all invertebrate model systems. So the big question is like, okay, does it translate into mammals and vertebrate systems though? So I'll just show you a very quick snippet of what it looks like in a vertebrate system. And we first turn to the turquoise killifish. And the reason why the African turquoise killifish has been this really big blowing up field in the aging field is because the the shortest vertebrate model system we had before the turquoise killifish was introduced was the mouse model, which has a lifespan of between like 3 to 3.5 years. So it takes a really long time and a lot of money to do a mouse aging study. The turquoise killifish is really interesting because it has a very compressed lifespan because it's found in Africa where these ponds of water only exist for about 6 to 9 months. So they need to live their entire lifespan in this 6 to 9 month period. And so because of that, they have a very short and compressed lifespan. Despite having this compressed lifespan, if you look at the trajectory of development to aging to death, it's very comparable to humans. So unlike it being like, oh, all the development occurs in a short window or a large window, and then lifespan occurs in a short window or a large window, the trajectory of development and aging in the killifish, where you see here in the bottom in the yellow, is very much analogous, where the development takes about like a 10 to 15% of their lifespan, and then adulthood is a majority of the lifespan. So this is a really nice model system because despite living only 6 months, the trajectory of aging mirrors what happens in humans. So we wanted to look at actin in the killifish model system. So we paired up with Berenice Benayoun, who is also at USC. Hengyu is her technician, who essentially collected all the killifish tissue for us. And then Aowen is my graduate student who did a lot of this work. So first, really quickly, we just did a very kind of generic RNA-seq analysis. So what you're looking at here is looking at fold changes between old and young. And so if you see a dot, it means that there is a statistically significant fold change in the gene expression of these specific genes during aging. Red dots means that there's an increase in expression, and a blue dot means a decrease in expression. And what you're looking at here is actin, ARP23 complex, cofilin, formin, tropomyosin, all those different actin regulatory proteins that I showed you at the beginning of the slide of the talk. And the only thing that I want you to kind of get here is that a lot of actin regulatory proteins, or I should say genes that encode for actin regulatory proteins, change in expression during the aging process. Not only do they change in expression during the aging process, but there is a lot of cell type or tissue type specific changes and sex specific changes. So if I give you one example here, like formin decreases in expression significantly in the spleen, but only in males. And then in females, there's no age associated change. In the male brain, though, it increases in expression, and in female gonad, it increases in expression. So there is both a sex dimorphic, that means sex specific, and tissue specific change in gene expression of actin regulatory proteins during the aging process, which is really interesting. And this is, of course, very zoomed out, looking at only at gene expression. So we need to actually look and see if actin organization and function itself changes in tissues. And I'll just give you one example. Here's looking at actin in the liver, and GRZ is just a specific killifish species. And you could see that actin in the liver, as actin should, make these really nice filaments. And these are the functional filaments. And in an old organism, you could see the actin turn into these balls, which is kind of reminiscent of those balls and aggregates that I showed you in the Drosophila brain earlier. So in the liver, essentially 100% of the actin become dysfunctional and turn into these big blobs of aggregates in old animals. And this is likely what drives failure of the liver with age. So that's all killifish work. We're also doing the same exact thing in mice, where my collaborator Peter Mullen collected all major organs that essentially matches the tabular mirrors. And Athena, who's my technician, with Gilbert, who's my lab manager, is now currently dissecting, staining, and imaging essentially every single major organ in the mouse throughout the aging process in both males and females to see how actin organization changes during general mouse aging. And I'm not going to show you any of that data right now because it's just a lot of image processing that we're still doing, and we haven't found anything particularly exciting to show yet. So just to show you some mouse data, I'm just going to show you some neutrophil data. And this is from a graduate student, Clayton Baker, who shared between my lab and the Benayoun lab. And for those of you who are not familiar with neutrophils, neutrophils are one of our white blood cells, and along with macrophages, are one of the most abundant immune cells. And they are part of your innate immune system, so they're non-specialized immune cells. So what neutrophils do is they kind of just circulate in your bloodstream. And when there is a infection, then specific cytokines will be sent out to the bloodstream to essentially signal for white blood cells to come to the tissue. So this is an SOS signal that goes out. Neutrophils will sense that SOS signal, go to that tissue, insert into the tissue, and then either undergo apoptosis, netosis, essentially what they're doing is just blowing themselves up to kill the bacteria, or they could phagocytose, that is engulf and eat the bacteria and then essentially destroy them inside of their body. So there are these like non-specialized cells that essentially will identify specific microbes and just kamikaze to kill the microbes. And this whole process is actin dependent, where what you're looking at here in this zoomed image is this is a single neutrophil that is not activated. And as soon as it sees a pathogen, it starts to polarize its actin cytoskeleton. So here in white is actin. So you could see a ton of actin filaments being formed. Not only are they formed, but they all get pushed to one side. And that's how the neutrophil will essentially go migrate to a microbe and either engulf them or kill them. So the entire functional process of a neutrophil, which is one to identify an infection and then kill it, is completely actin dependent. So this whole process is actin dependent. And so what we can do is actually quantify the amount of actin that a neutrophil synthesizes upon seeing some kind of infection. So in vitro, we stimulate the neutrophils using LTB4 or some kind of cytokine, essentially. And you could see that upon seeing a cytokine, neutrophils will increase the amount of actin it has very, very, very rapidly within 60 seconds. And this stays on high levels of actin for about like 30 to 60 minutes, and then eventually it'll resolve. In a young neutrophil, which is this light pink line, you could see this very robust response where actin filaments are synthesized. And then in old neutrophils, you see that they actually don't have the ability to create actin filaments. And this dysregulation and this inability to synthesize actin filaments is what drives neutrophil failure with age. And again, this is still correlative evidence, so we need to show causative evidence. So we need to show that if you break actin, you will make these neutrophils fail. So to do that, we use a drug called latrunculin. What latrunculin does is it binds to those monomers of actin and essentially prevents them from polymerizing. So here, what you're looking at again is looking at the ability of the neutrophil to undergo chemotaxis. That is, it builds those actin polymers to essentially move towards the site of infection. Young cells are very much able to do that. Old cells are not. But if you treat young cells with latrunculin, that is, you destabilize actin and you prevent actin filaments from forming, they basically fail. So they're mimicking those old cells. The actin dysregulation doesn't affect old cells because they're already dysregulated. But essentially, just by disrupting actin, you can make otherwise young and healthy neutrophils behave like an old and dysregulated neutrophil. So essentially, what I've shown you so far is that yes, actin deterioration occurs naturally during the aging process. Disruption of actin can induce phenotypes of premature aging, and this all happens in vertebrates. This happens in non-invertebrate model systems. And then I'm just going to show you a very quick snippet in humans, where this is looking at actin in the human brain, and this is just visualizing with using phalloidin, and you can see those really nice organized filaments of actin. And then if you look at an Alzheimer's disease brain, you can see these massive aggregates of actin forming. This is very reminiscent of those actin aggregates that I showed you before in Drosophila brain during natural aging. So during age-related pathologies such as Alzheimer's, you can get very, very, very similar actin aggregates form. And these actin aggregates are essentially inducing neuron degeneration and neuronal death. So right now we're trying to figure out what are ways that we can try to resolve some of these aggregates, basically the methods that were used in this fly paper, and see if we can apply that to a human neuron. And by reducing the aggregation burden in human neurons, could we essentially mitigate the aging process? Okay, so I've shown you so far, yes, actin deterioration occurs during natural aging, and then directed and targeted disruption of actin can induce premature aging. And then, of course, the big flip side is, well, can you protect actin filaments? Can you prevent them from breaking down? And can you mitigate the aging process? And so, can we target actin for anti-aging therapeutic intervention is like the big question that always comes up, right? So, essentially, we know that actin breaks down during the aging process. Can we reverse this breakdown process? I'm going to skip a few slides here because I thought I took these slides out, but I didn't. And I want to get to something else that I think is more important. So one of the easiest ways that we could stabilize actin is I've already shown you that if we destabilize actin and disrupt actin using genetic methods, we can reduce lifespan. So the other direction is to stabilize actin, essentially, basically prevent them from breaking down during the aging process. And can we extend lifespan? So I paired up with a marine natural chemist, Tyler Johnson. What he does is he essentially isolates naturally occurring chemical compounds from marine organisms like sponges and coral. And so one molecule that he isolates is called jasplakinolide. And what jasplakinolide does is it directly binds to filaments actin and stabilizes it. So we use jasplakinolide. This is in worms. And you can see that low concentrations of jasplakinolide significantly extends lifespan. If you go too high, you can actually reduce lifespan, because one thing that I should say about actin is that it's very dynamic. So if you over-stabilize actin, it can be toxic. But if you can find a really nice window where you can stabilize actin just enough, you can have a significant effect on lifespan. So yes, you can kind of fine-tune actin in both directions, where breaking it can reduce lifespan and stabilizing it can extend lifespan. Now, because the stabilization is a little bit finicky, where you need very, very, very precise concentrations to make sure that you're having a beneficial effect and not having a detrimental effect, when we think about therapeutic translation, this becomes a little bit scary to think about, because if we give you too much, you could die. So we don't like therapeutic interventions like that. So when we think about protecting actin filaments to mitigate aging, yes, you can do it, but we need to find a better way than just using a drug that's going to stabilize actin, because that can be toxic. So then, in kind of thinking about how actin regulates many different processes within the cell, we started to think about, well, what are some of the processes that actin regulates? What are some of the cell components like organelles that actin regulates? And can this be bidirectional? So what my lab has started to do is understand how actin can regulate mitochondria, nucleus, and endoplasmic reticulum, lysosome, other organelles, and then how the organelle can also feed back into actin. So I'll just show you a very quick snippet of how mitochondria and actin can have this like really intricate crosstalk between each other. And the reason why we really like mitochondria is because our lab studies stress response, and mitochondrial stress response is very, very, very heavily studied. As you all know, mitochondria, people call it the powerhouse of the cell. We all know mitochondria have many other functions in energy homeostasis, metabolism, apoptosis, etc. But one of the primary functions that everybody knows about for the mitochondria is energy production through the electron transport chain. So you have complex 1 through 4, which pump protons out into the intermembrane space. And when they come back in to the matrix, you synthesize ATP. And the reason why we really think about the electron transport chain and care about it is because a lot of stress biologists have found that if you disrupt the electron transport chain just a little bit, you can do something called hormesis. So what is hormesis? It's this concept that what doesn't kill you makes you stronger. So a little bit of stress can be good for you. So if you stress out the mitochondria by electron transport chain inhibition or any other targeted disruption of the mitochondria, but you just do it a little bit where you're not blowing up and destroying the mitochondria. You're doing acute mild stress to the mitochondria. You can actually activate a beneficial stress response pathway, which we call hormesis, and that can extend lifespan and health span. So this would be like if you exercise. When you exercise, you cause stress to your body, your heart rate goes up, your body sweats, you get micro tears in your muscle. You actually induce a little bit of mitochondrial dysfunction by applying too much stress to the mitochondria. But we all can agree that exercise is a good thing. So this little bit of stress can activate a very robust and beneficial response. And this can actually extend lifespan. So it's been shown I'm showing you a C. elegans data here, but it's been shown in C. elegans, it's been shown in mice, it's been shown in yeast and Drosophila, that a little bit of mitochondrial stress activates this beneficial mitohormesis process, which can massively extend lifespan. And so, of course, the question that I asked was, we know that if you apply actin dysfunction, it can reduce lifespan. We know that if you do mitohormesis, it extends lifespan. So my postdoc, Norbendo, was like, okay, what happens if we just add these together? Normally, if you have like a negative and a positive together, if you add them together, you just get a net neutral, right? So if there's no synergistic effect, if there's no compounding effect, if there's no protective effect, we'll just expect the combination to hit zero. But what was really exciting and cool for us is actin dysfunction reduces lifespan, mitohormesis extends lifespan, but when we put the two together, it's actually just as long-lived as the mitohormesis itself. What this suggests is that maybe mitohormesis can have a protective effect against actin dysfunction. So we looked at that through a little bit more targeted method, where I showed you before that if you apply actin dysfunction, you cause these massive fragmentation of mitochondria. And if you apply mitohormesis on top of that, you can actually restore mitochondrial morphology. Not all the way to wild type, but that's because mitohormesis itself causes a little bit of mitochondrial fragmentation. But you can have a pretty significant restoration of mitochondrial morphology. We could look at something completely unrelated, like protein aggregates. So here you're looking at polyQ, which is an aggregation-prone protein. And when you induce actin dysfunction, you massively increase protein aggregation burden. And when you apply mitohormesis on top of that, you completely eliminate these aggregates where you're really protecting against actin dysfunction in multifaceted ways. So you're protecting against the lifespan dysregulation, you're protecting against protein aggregation, you're protecting against mitochondrial fragmentation. And we looked at a whole slew of phenotypes like health span measurements, other metrics of cell biological hallmarks of aging. And it seems like mitohormesis protects against actin dysfunction in a multi-model system. So this intricate mitochondria to actin kind of connection really makes us think about, could we apply this concept of using mitohormesis to protect against actin dysregulation as a novel anti-aging intervention where mitohormesis methods are actually being applied even in humans now. So one really good example is metformin, which is a mild complex 1 inhibitor. And people are really obsessed with this concept of metformin being an anti-aging gerotherapeutic drug. And so we're kind of coming in from a different molecular angle that people haven't thought about before, where if we could use mitohormesis to mitigate anything associated with actin dysregulation. If the answer is yes, then all the things that I just showed you, general aging, Alzheimer's disease, neurodegeneration, muscle myopathies, even cancer, all the things that occur because of actin dysregulation could potentially be restored and targeted by using mitohormesis. And so we're kind of doing a copy and paste of this mitochondria actin connection with the endoplasmic reticulum, with the nucleus, with the lysosome, and all the other cell components to see if there's any other bidirectional regulation of actin to a specific organelle or cell component. So I'm going to end there so that we have time for questions. So this is my whole lab. I think I thanked everybody as I kind of went along, but these are all the great people that make the magic happen. And here are some of our collaborators and our funding sources. And with that, I'll just stop now and thank you all for listening.
Thank you, Rio. This was a really wonderful talk. And acting, I think, as a biological hallmark, it's a very exciting perspective for aging biology. I wanted to remind the audience to add their questions to the Q&A section again. There are already a couple of questions while the others are coming. I just want to ask, you show data across the species. So across these like different model organisms, do you see a conserved cytoskeletal aging signatures, or do you think these mechanisms are more species specific?
Yeah, that's a really great question. I think when we just look at actin like organization, it's probably going to depend on the tissue. So the muscle is probably where like aging pathology and aging like hallmarks are shared the most in terms of actin, because I think actin regulation in the muscle itself is just very, very, very conserved. And how actin works in the muscle is very conserved. So when we look at actin breakdown in the muscle, that's very similar. When we look at things like neurons, where neuron aging across species is a little bit less conserved, especially from invertebrates to vertebrate systems, where even the ratio, for example, like glia versus neurons is very, very different across model systems. That's where like things are a little bit less conserved. But it is still interesting to see, despite the lack of conservation and brain aging, we do see that actin aggregate seems to be the driver of aging in the brain.
Mhm.
So I say yes and no.
Yeah, I
Yeah.
agree. So I move on with the questions in the Q&A. Larissa says, very interesting talk. Considering that aging is a normal process, is it correct to use phrase aging pathology? I think this pathological connotation may unintentionally put older into negative mood.
That's a great question. Yes. You're right in saying that, like, yes, aging is a natural process. So should we call it aging pathology? It's definitely like a semantic argument where like people want, I think that like at one point NIA wanted to call aging a disease because calling something a disease was really the only way to convince Congress to give them funding, right? But NIA also recognizes that calling aging a disease is a bad thing because we don't want to call natural healthy aging a disease. I think right now it's just a political thing that we call it a pathology because that's the only way to convince people to give us funding to study aging. And to call it a pathology calls it something fixable, because telling Congress that something is fixable and our research can find a way to fix it and extend lifespan is the only way people give us money. And this works for donors too, where like a lot of people, a lot of philanthropists want to hear that aging is something that can be fixed. So we need funding to fix it. But I completely agree with you that as like a biologist or really as a human, we shouldn't call it a disease. It's not a disease at all. It's just what happens naturally.
Yeah. Thank you. Another question from Saidas. Excellent talk. When you destabilize actin, is the pattern of changes in actin disruption, weakening, the same as how it occurs during the aging process?
In some instances, yes. So for example, if we disrupt tropomycins and that essentially destabilizes actin, because what tropomycin does is it binds to and stabilizes it. So when you get rid of it, you just destabilize actin. That is pretty similar to what happens during natural aging. If you block something as critical as the ARP23 complex, I don't think that that's mimicking aging. Like disrupting the ARP23 complex, which is important for synthesis of actin, microfilaments and like the branch actin networks would be the same as like hitting somebody with a truck. Like that's not mimicking natural aging. So yes, I think that if you do like fine-tune disruptions, yes, that's mimicking aging. But when you do these like large scale, big hammer approaches, it's probably not mimicking aging at all. But sometimes, like but why do we do those methods? I think when we do these big hammer approaches and we can find an intervention that protects against that, then likely that intervention will protect against aging, which is a much milder dysregulation of actin. So those like mega hammer like approaches to break something might seem like physiologically irrelevant, but if you can find an intervention that can fix something that dramatic, then it gives us a little bit more hope in terms of like targeting diseases or aging.
Next question is from Arpit Sharma. Thank you, Rio, for such an insightful talk. Besides mitochondrial dynamics, are there alterations in mitochondrial bioenergetics, metabolism, or membrane potential in animals treated with various actin RNAs? Perhaps even how organelles are trafficked.
That's a really, really, really good question. We haven't looked at trafficking. It's a little hard to do in worms. I know you're a worm person, Arpit. So if you know how to do trafficking in worms, I'd love to do that together. We've looked at respiration. Respiration is significantly lower. We're doing the metabolomics right now. I assume it's going to be different just based on how badly organized the mitochondria are. Right now, we're working with the Luger lab at UCSD to make ATP sensors, both mitochondrial and cytoplasmic ATP sensors. So we'll know for sure what ATP generation looks like in these actin dysfunction animals. So far, we only looked at morphology and respiration. They're both significantly dysregulated. But yeah, I'd love to look at trafficking and more like metabolite specific things. We're doing the metabolomics hopefully in next month. We're collecting the worms right now.
We have another question from Saidas. Are there ethnic populations or other phenotypes that have superior or inferior actin organization at development, especially for the muscle?
That's a great question. We haven't looked at diverse populations. It's a little bit hard to collect the data because anybody who does population health or epidemiological studies will know that most of the data is collected in affluent white individuals, but there are data sets that are starting to like be collected now. There are specific like indigenous populations where a lot of these studies have been done. So we have some collaborators that are looking at indigenous populations, especially ones like that are protected from specific diseases. Like there are indigenous populations that have much lower incidence of Alzheimer's disease despite being a developing nation. And so we're kind of mining those data sets to see if part of that could be because they have better regulation of actin. So it's a really great question.
Yeah. And Thomas Mordy asked, does actin destabilization slow down after target grades exit cryobiosis?
Great question. We're doing that right now. So we just basically built the tool. It took us 3 years to basically figure out how to like synchronize tardigrade populations, figure out how to do a lifespan in them, and figure out how to like age them out reliably. And so now we have those tools and we kind of have started to define what actin aging looks like in tardigrades. So now we're putting them into cryptobiosis and like there's a lot of questions, right? So it's does cryptobiosis rejuvenate animals? Or does cryptobiosis, is it like a diapause state where they stop aging? Or, do they just continue to age in cryptobiosis? We have no idea. So we're going to test all of these things.
And Jean Donwan, great presentation, thank you. While mitohormesis could rescue actin function for anti-aging, but in terms of cancer, can this actually be detrimental, promoting aggressive cancer growth and metastasis?
That's a great, great, great question. And I love that question because that's all the cancer people ask me that question. So one of the great things about mitohormesis actually is that while it's beneficial for non-cancer cells, it's actually either detrimental or no effect on cancer cells. So we've done a lot of profiling of cancer cells, and if you activate mitohormesis in cancer cells, it either will induce apoptosis because they're very energy sensitive. So when you do any kind of dysregulation of energy homeostasis or mitochondria, it tends to be bad for cancer cells. And for some cancer cells, it doesn't affect them. But the metastasis question is a fantastic one because the reason why people don't do a lot of mitochondria targeted cancer therapies is because they have a tendency to actually increase the risk of metastasis. Now, the reason why actin mitochondria together is such a beautiful thing is that when you disrupt actin, you stop metastasis completely. Actin is absolutely essential for the entire metastatic process. That is, the sloughing off of cells from the main tumor, going through the blood circulation, and then adhering at the new site is all actin dependent. So, actin disrupting drugs are the number one most effective cancer therapy, but they're also the most toxic because I just showed you how actin disruption kills everything, right? So what we're trying to do actually now is could we use mitohormesis as a protective effect where it's going to shield your healthy cells from anti-actin drugs and then go in with a very heavy dosage of anti-actin drugs to kill and wipe out your cancer cells while essentially having a 0% chance of metastasis? And can mitohormesis keep you healthy long enough for you to survive an actin dysregulatory drug? So we're thinking about it in terms of a combinatorial treatment for cancer. You definitely cannot go in with mitohormesis alone. It's been shown time and time again that that's not going to work. And same thing with actin dysregulation. But if you can use both and that works synergistically really well, then that will be a nice therapeutic strategy.
And Chris Wiley asks, lovely to finally see your talk, Rio. I was curious about the idea of chicken and egg relationship between mitochondria and actin. The actin disruption of mitochondria was compelling, but if it doesn't get to the point of complete disruption, do you think there might be a sweet spot where a small amount of actin disruption might drop mitohormesis itself?
Mhm. That's a great question. I wanted to find that. So we did do very, very, very mild dosage and we tried to titrate as low as we possibly could. And we could never find a dose that induces hormesis. I think the problem is that all hormetic methods are transcriptional and that a little bit of stress activates a transcriptional stress response. And that is what drives the beneficial effect. And right now, I have an entire story that one of my graduate students is working on that actin is actually required for transcriptional regulation. So when you disrupt actin even a little bit, you're actually reducing transcription. So then you can't activate stress responses. So I don't think actin hormesis will ever be a thing. In terms of specifically mitochondrial stress response, the chicken and egg thing is really interesting because we've seen in our lab that actin function is required for activation of mitohormesis and mitohormesis is required for actin regulation. And I can't figure out which one comes first. Like, I don't know how to do that. We've tried because it just seems to go in both directions. So if anybody has ideas, I'm very happy to talk about it.
Yeah, that almost happened in all cases during
Yeah, yeah,
we study some questions. And maybe we can take one final question if your time allows,
yeah.
Rio. Sayede does ask, are there autoimmune disorders that disrupt actin more like the sledgehammer insults?
Yeah, that's a great question. There are a lot of autoimmune diseases that are actually caused by, not caused, I shouldn't say caused, that are associated with dysregulation of actin in immune cells. So I think a lot of the immune work that we're working on with Benyun Lab, I showed you just the neutrophil work, but we're looking at macrophages, we're looking at adaptive immune system as well. A lot of autoimmune diseases are caused by dysregulation of the innate immune system as well, which is very heavily ignored, which is why Berenice and I are focusing more on innate immune cells. There's plenty of people working on T cells and B cells with actin regulation. So we're really excited about like looking at how neutrophil and macrophage dysregulation can be causative drivers of like aging and autoimmune disease and other diseases and how actin plays into that. So we have like a whole R01 that we just wrote looking at neutrophils in that specific pathway and how it's sex dimorphic because autoimmune diseases are also sex dimorphic, where women tend to be at higher risk for autoimmune diseases at younger ages and then it flips in older individuals.
So thank you very much, Rio. This was a really exciting presentation, and thank you to everyone who joined and participated in the discussion today. I think we are over time, so have a great day.
Great. Thanks so much, everybody.
This was